All right, I guess it's pretty close to start time. So my name is Cole Edwards. I teach at Appalachian State University in Boone, North Carolina, United States. And I guess I'll be moderating the morning session here. And so I just want to thank everyone for coming, welcome you to day 3 of what hopefully is another excellent day of these Zoom presentations for the IGCP project 653. I just want to remind you that this is recording, so we'll post this to the YouTube channel stream later. What else? Yeah, so we'll kind of operate in the same fashion where talks will be approximately 15 minutes. We'll save any time thereafter for questions. The speaker may not be able to see whether you're raising your hand or even if you're using the raise hand function on the Zoom. So perhaps you could write something in the chat and then I can see whether or not you have a question, and I can at least draw their attention to your question, and you can ask it either yourself or I can relay the question if you choose not to. I think that's been working pretty well so far. I guess please save the chat, be judicious and appropriate with your comments. I guess Christian will play chat police if we have to get that far. And then of course, please abide by the code of conduct that has been scripted and provided for us. So hopefully we have a great setup of talks coming up. We have the morning session and then the afternoon here, at least afternoon for me in the States. So I believe with that, we'll start with the first talk. So we have, first talk is gonna be given by Jana Bruthansová and her colleague Heyo van Iten. This is a recorded talk, and unfortunately Jana won't be able to attend it to answer questions. So we will, I guess, just play the clip, and then we will transition to the talk following hers.
Dear colleagues, let me to introduce you our research focused on Ordovician conulariids from Bohemia, which served as host organisms for diverse epibionts. My name is Jana Bruthansová, and I am working in the National Museum in Prague. And my co-author Heyo van Iten is teaching at the Hanover College in Indiana. We investigated conulariid material from the Upper Ordovician of Morocco, and mainly from the Ordovician of the Czech Republic, which is located in the middle of Europe. Elongated Prague Basin extends about 110 kilometers from just east of Prague to the city of Pilsen. And it's now denudation relict of an early mid-Paleozoic rift basin formed on the Cadumian rocks. Sedimentation started in the Lower Ordovician and continued with no interruptions to the Middle Devonian. The Ordovician section measures up 2,500 meters thick and consists of 12 formations, collectively ranging from Tremadocian to Hernantian in age. Conulariids first appeared in the Prague Basin in the Lower Ordovician, and their diversity gradually increased to a maximum of 14 species in the Middle Ordovician Ceara K.Q formation. The last occurrence of conulariids in the Ordovician of the Prague Basin is in the Upper Ordovician Králův Dvůr formation. Let me to introduce you the conulariid morphology, and their systematic position. They are supposed to be extinct Scyphozoan cnidarians, widespreadly ranging from the latest Ediacaran to the latest Triassic. The conularid polyp was largely enclosed with a steeply pyramidal, organic or organophosphatic periderm that varied in surface relief from nearly smooth to strongly corrugated. Conularids attached to the biological substrates at the apex of skeleton, which may be broken to the rounded structure called shot. Ordovician strata of the Prague Basin in the Czech Republic contain some of the most abundant conularid faunas in the world. The number of Ordovician conularid specimens reposited in major museums in Prague and Pilsen approach several thousands and continues to grow as classic collecting localities are resampled. Some of these conularids exhibit epibionts, including brachiopods, bryozoans, and adrioasteroids. Together with Hall's monograph from the year 1852 on the Devonian fossils of New York State, Baran's monograph on conularids of Bohemia from 1867 contains the earliest published illustrations and description of Epizoic adrioasteroids on these fossils. Bouček's monograph on Bohemian conularids from the year 1928 builds on that observations. As I told, our research focused on diverse Epizoic organisms which used conularids as the host substrates during Ordovician. We did the first comprehensive survey of epibionts on Ordovician conularids from the Prague Basin and identified and enumerated the epibiont taxa and their conularid substrates. We focused to the distribution and orientation of epibionts on the conularids and their spatial relationship to each other, the stratigraphic and facial distribution of the conularid epibiont associations, on the taxonomic diversity of epibionts on individual conularids, and to the size ranges of the epibiont taxa. Based on these observations and measurements, we discussed the timing pre- or post-mortem of attachment of the epibionts to their conularid substrates, as well as the bearing of our observation on alternative interpretations of the mode of life and life orientation of conularids in general. We also compared the Bohemian material with other substrate groups in the Prague Basin and with encrusted conularids recently documented from the Upper Ordovician of Morocco. We found that approximately 5% of just over 5,000 specimens exhibit invertebrate epibionts and or attachment scars. The most commonest epibionts are brachiopods, followed by bryozoans and adrioasteroids, which are invariably articulated. Less abundant are monoplacophorans, Sphenothalus lichenocrinus holdfasts, and other undeterminate invertebrate holdfasts. Epibionts occur on the species from open shelf deposits in the Upper Ordovician Letná and Zahořany formation, which conularids are substantially more abundant than in other formations. Here you can see epizoic brachiopods, mostly highlighted by arrows. Note that brachiopods are absent on the strongly protuberated apical region called shot, marked here by white arrows. Epibionts on particular conularid belong to a single species or up to 3 species and major taxa. Except for monoplacophorans, epibionts occur on the external surface of conularids, in some cases with brachiopods, bryozoans, or adrioasteroids present on all 4 faces. Brachiopods and adrioasteroids present on all 4 faces show little variation between the faces in their size range. You will see it on the next slide. Also, brachiopods show apparent random trending of the antero-posterior axis, marked, for example, here by dark arrows. We found terpostome and cystoporate bryozoan colonies encrusting the external surfaces of some specimens. In some cases, all 4 faces, including the apical region. 18 specimens of this adrioasteroid encrusted all 4 faces of this conularid skeleton. Note that the adrioasteroids are centered closer to the facial midline than to the corners. We also note that even though the adrioasteroids vary in diameter, the size range of them on any one face is similar to that of the adrioasteroids on the other 3 faces. The same observations we did on the Moroccan material. Monoplacophorans were mostly originally attached to the internal surface of conularid, now preserved as an internal mold. The latest table represents photos of conularids with diverse attachment discs of Sphenothalus and other holdfasts. These results highlight the importance of conularids as biological substrates, especially in Perunica and South Polar Gondwana, and indicate that incrustation occurred both on live individuals and on dead ones. The corners and midlines influence the settlement and subsequent growth of brachiopods and adrioasteroids, and conularids bearing these epibionts were buried catastrophically. This specimen probably represents conularid with fall down and then was covered by diverse epizoic brachiopods. Terpostome, bryozoan, encrusting specimens of Archaeoconularia fecunda, very small conularid, may have overgrown these specimens in its original erect or semi-erect life position. Finally, conularids exhibiting monoplacophorans on their inner surfaces and these bryozoan colonies which covered all 4 faces of conularids and also adapical region probably were encrusted after that. Related to the Great Ordovician biodiversification event, we really can observe in the Bohemia huge increase in the conularid diversity from 2 in the Lower Ordovician to 14 in the Darwinian, although the position of Prague Basin was in that time in high latitudes on the southern hemisphere. Their diversity up to the younger Ordovician strata gradually decreased. The same was also in the Silurian and Devonian, when Prague Basin was located in the subtropical latitudes. This was conularid species diversity, but what about their colonization? According our datasets based on Bohemian and Moroccan Ordovician specimens, the record of conularid epizoic organisms indicates that the increase in their diversity during the initial stage of GOPE was very low, related to the Ordovician radiation in the sense of Stigall et al. 2019, we can observe the rapid epibiont diversity and also abundance increasement during Sandbien and Katian. Due to the family care, this presentation was pre-recorded and I am not able to respond to your questions in this moment. But if any, do it please by chat or email later. I'm sending you my best regards and also greetings from my youngest daughter, undoubtedly future paleontologist too. Thank you for your attention.
All right, great. Yeah, apologies. If you have any questions, you know, I guess we can throw them in the chat if Jana can join us later, if she's not already here. Or, yeah, simply send an email. Okay, so it looks like I guess we're a little early, but these tend to go long. So maybe if Ben, are you ready to put your slide up here? And while you're doing that, I can introduce you and your talk.
Yeah, working on it just a bit there.
No problem. We've got a minute here. So Ben Dattilo will give a talk titled "Tracking the Water Depth Habitat Range of Megalograptus through the Katian from the Appalachian Basin to the Cincinnati Region." So Ben, feel free to take it away.
I think you're muted, Ben.
I found it. Can everybody hear me now?
Perfect.
Much better.
Very good, very good. Oh, I'm having a old man time. Okay, so everybody can see it. I don't know how that red square got there. But basically this talks about Eurypterids, but specifically Megalograptids, and the reason we're doing this is because Eurypterids are cool, and Megalograptids are even cooler because they're really weird. Now, so what we're going to cover is what happened is Plotnik approached me about trying to figure out what the paleo environment was for these things, because Carl Brett and I had recently finished a paper on the stratigraphy of the Cincinnati region, where they're very common. But as a warning here, there's a lot of bias in these things. You imagine when you see a megalograptid, or when you think about these, you think about these fully reconstructed things. But in reality, they are little scrappy bits. They are very hard to recognize, especially by the casual collector. This particular Mount Orab occurrence, for example, was collected, is just a few specimens from years of shale digging for trilobites by a person who kind of knew what he was doing. Otherwise, I think that they're missed. So anything I say And this is the specimen that, upon which the original drawing here was made. So this original drawing was based on bunches of specimens found in one locality in the early 1900s and or in the mid 1900s. Otherwise, what you get is these scrappy bits. This is LaGrange, Kentucky, and you can imagine that people just don't generally notice them. Therefore, the data I present is sparse enough that we can make any story we want to, which makes it even more fun. So, just to talk about where these things are occurring generally, note that they are in the Ordovician. I've circled the Ordovician colors here. They are pink on this map of just the United States. We do have some up here in Quebec as well. But the outcrops are also spotty, which makes it even easier for me to make up any story I like. But we have them here along what were the tectonic origin. And then they're here in the Cincinnati region, where they're very abundant. Whoa. The earliest ones found up here in Minnesota. And there are other Ordovician outcrops that you can see where they have not been reported. We don't know if that's bias or if that's real. So here are the occurrences. And I've divided this into the continental arch, which is the earliest one, which is a really bizarre occurrence in the Winneshiek Lagerstätten in a crater, basically, during the Darwinian. Then they show up in the Appalachian Basin, so these colors here, and notice that this order is not specifically, I just had to fit them on the diagram. The stratigraphic interval for these things is basically, we are about 80% sure they occur within this slice right here, this orange slice, about 80% that all these occur within this yellow slice. And again, that brings us up into the upper Katian slice 1. And then as we move into the Cincinnati region, we don't find them anymore in the Appalachian Basin, even though there are rocks there. And we don't find them earlier in the Cincinnati region, even though there are rocks there. So we're going to try to argue that this probable bias is actually data in this talk. And what we see here is that there's also a lot more occurrences as you go up the section into the last of the Katian. Possibly this elkhorn is actually Hernandian. That's still a little bit debatable. But you see that there are bunches of occurrences, and I had to just cram them in on this diagram. So I'm going to go through a map here. On the right here, you see a star just marking where this is stratigraphically. And then on the map, this is where it is, where the actual spot is. So this is the Winneshiek Lagerstätte, an occurrence then in New York. And then several occurrences up by the Edenian or Katian 1. And they seem to spread out from what used to be the tectonic range right here. Then they make a jump across the covered interval. This is mostly Pennsylvanian, coal measures and stuff. And the earliest one is in Kentucky, as close to the Appalachian Basin as you can get. Then they start moving north and a little bit west. That one's odd. We'll get to that later. Also notice that things are odd down in this region. And then they move back generally to here. So we'll zoom in on that in a minute. And the overall story that you kind of get a sense of is that perhaps these things are actually prograding with the Appalachian fill. And so that this migration and invasion of the Cincinnati region may in fact be tracking the facies, a particular facies. But what facies that is a question. Of course, in 1964, Castor and, I'm sorry, I can't pronounce that name. I'm sure some of you can. Basically was under the impression that everything was layer cake and didn't really understand facies. So the big question that he addressed in describing these Megalograptids was whether or not they were marine. Now, since that point, we've taken, I guess I'll back up here and say that mostly these things are found in mudstones, and since that point, we've spent a lot of time, especially Carl Brett and others, on working out both the fine scale time slice stratigraphy as well as the details of facies change that are not really very apparent just from looking at the rocks. For example, here where you don't find any Megalograptids, in the lower part of the Cincinnatian, the approximately Katian 2 or 1, is deeper water fauna, very small things, really cool stuff here, but again, not so cool as a Megalograptus. As you move up, here's the Mount Orab Megalograptus, where the trilobite collector has been working for years and years and found a few scraps. You notice a very different kind of fauna. And if we just compare to today, for example, these ribbed mussels are definitely today a shallow water fauna. And so we've worked out layer or time slice by time slice, what is shoal and lagoon, what is shallow subtidal, what is deep subtidal, and what is offshore. And this is one particular time slice in the immediate region of Cincinnati. And in this time slice, it's just important to note that this is one time slice, and these, whether it's lagoon or shoal or shallow shelf, you notice that it's roughly to the north-northwest that it gets deeper. So if we track these things, here's approximately 5-meter bathymetric lines. And we start with the first occurrence, which is again down here. The second occurrence, it's really helpful when there's only one dot. All you have to do is say, oh, that's obviously moving northward. There's another dot, there's another dot, and then we start getting in, oh, okay, so this was collected in the 1980s, and oddly enough, in the Cincinnati region, the stratigraphic refinement of the early work of Castor was far better than anybody in the 80s. So I suspect that this weird backwards tracking might actually be a stratigraphic problem in identifying where this thing is. Then there's this one, and what you notice right here is up here is where there are 3 localities, and then down here is another locality, and this was collected a long time ago. It's a little iffy down here because this was actual facies change in the Cincinnatian, and I don't think Castor and his previous people that worked on the Cincinnatian quite got facies and may have been just tracking the same facies and thinking they were in the same time slice. Normally they are pretty good. You notice I'm just sort of explaining away the weird points, which you'll allow me for now since we only have 3. Now, also, this is like 200 meters from my childhood home, and so we expect it to be a little off. And then down here, in the Elkhorn, again, this is the original right here on the right, is the big occurrence that was written up by Castor and is the basis for the entire reconstruction. And it roughly roughly tracks all the way across to the other side of the outcrop. In the middle, we don't have anything that age, by the way. It's all eroded away. It's older. And again, we have this point that's very poorly documented in my hometown. At any rate, if we look at all these together, there is a rough tendency for things to move north and west a little bit, and then suddenly go back down again, which kind of matches the work that we've done. So what I've done here is what I show on the left is a recent, from 2 years ago, I believe, the IGCP, we presented this paper where we show shallow water to the right, that's south, and deeper water to the left. But what's important is that the facies advance. You notice that in the Cope Formation, for example, where we find no graptolites, or sorry, Megalograptus, that we also have the deepest water, but the shallow water environments advance upward. There is an overall progradation with a little retrogradation at the top, and this matches our occurrences of these Megalograptids. The conclusion from this would be that basically these things are tracking just the shallowest of the subtidal facies and can be found possibly in even shallower deposits. So now we are better than just they are normal marine. They're in shallow subtidal marine. On top of that, this is mostly Roy's work, is the question of what did they eat, what were they doing in the shallow marine? And we'll see a lot of reconstructions of them attacking our ancestors or our near ancestors and causing a lot of havoc that way. And you just over and over, this is the Winishik reconstruction, and again, they're attacking our ancestors, they're attacking fish. But in all seriousness, fish had teeth for skin. And these things, I love this reconstruction by Takashi Ito, made of paper, which makes me think of just how delicate these spines might have been. So these are little bitty spines coming out, and you're trying to attack something that's armored with teeth. And here's an actual fossil again showing just how delicate and thin these are. And even worse, if we look in the Cincinnati, where we have a whole bunch of these things, there's a little problem in that there were no fish. There were conodonts, but those aren't the fish that, that are reconstructed in these pictures. So we have abundant Megalograptids and no food. I mean, abundant as they get, right? They're scrappy and rare. So again, this is a lot of Roy's work. What we've found, what's been coming up lately is that jellyfish are very yummy and nutritious. And I work a little bit with Frank Palladino, who's been tracking leatherback sea turtles forever. So I stole this from his Facebook, and he likes to call pyrosomes jellyfish, which just bothers me. But from a food standpoint, they are jelly-like animals that float around in the ocean. So they are jellyfish, I guess. At any rate, you notice that the throat of the leatherback is full of these kind of soft, sharp, backwards-facing spine things. apparently adapted specifically for eating jellyfish. And if we look back at our Megalograptids, we have these soft spine-like things, and what this picture shows here, I forgot to cite this one, but it's a paper. Basically, is if we want to capture jellyfish in the deep sea, we've got to do something like this. So this is a little invention somebody made to catch jellyfish in the sea using little remote vehicles. So, in conclusion, we are going to stand by the conclusion that Cincinnati and Megalograptids are found in shallow subtidal deposits and ignore any possible bias on that for now. They appear to have invaded from the Appalachian Basin, and they might have simply followed the environment in the region, basically sticking to the shallows as the facies prograded. This is at least consistent with jellyfish hunting. Thank you.
Yeah, great talk, Ben. I think we have time for a few questions if people want to just either pipe up or post your question in the chat and I can relay it. Guess I'll selfishly take this time and ask, Ben, do you ever see evidence of, of the few specimens that you have with the spines preserved, do you ever see evidence of damage and or regrowth or healing of these?
That would be a Plotnik question. I don't actually study the critters. I'm the stratigrapher.
No,
Roy's here.
yeah.
Yeah, he's in the Ordovician.
Right. The specimens that I've looked at, again, the spines are rarely preserved. That's it all. And there's
Yeah, Axel.
Why can you exclude conodonts as prey?
Roy?
That's why didn't they eat conodonts? Too small? Not.
well, possibly they could have. Again, the basic argument I'm going to be making here is that the large, thin spines would have broken with anything that had any muscles, and so it's more likely that and the design of the appendages, I'm going to do this. It's a trap. It's a trapping mechanism rather than a grabbing mechanism that they have there. And again, that seems to be consistent. There are certainly a lot of jellyfish around, and again, recent studies have said they're much more nutritious than we've thought previously. So this is we're just sort of saying, let's expand get away from the idea that these things are these fierce predators that are biting on muscular things or biting on shelled things, but they could be trapping things that are floating in the large things floating in the water.
Thanks.
We'll take one last question. Javier had a question about whether there are any known examples of modern arthropods predating on jellyfish.
Not that I'm aware of. There's a lot of snails that predate on jellyfish. There are, I think there may be some shrimp that do. Certainly the sea turtles are an example of that.
Okay, great. Well, thanks so much, Ben and colleagues, for giving a great talk. We'll move on to our next talk here. We have Ceara Losso. I think I saw her name. Yeah, if you want to throw up your slides here. So Ceara and Javier Ortega-Hernández, they'll give a talk titled Recent Developments in the Paleobiology and Taphonomy of Trilobites from the Walcott-Rust Quarry, Upper Ordovician. So Ceara, take it away.
Thank you. So I'm a PhD candidate at Harvard University. working broadly on early arthropod evolution, and today I'll share some of the work which is on the paleobiology and taphonomy of the Walcott-Rust. The quarry is located in upstate New York along the West Canada Creek near Trenton Falls. And this quarry was made famous by the work of a very young Charles Doolittle Walcott when he was like 11 to 20, when he published results showing trilobites with preserved appendages that were actually preserved in calcite. In the quarry, there are two formations exposed, the Rust Formation and the Steuben, and the specimens I'll be showing today are from layer 3 of the spillway member of the Rust Formation. These layers were deposited in the Mohawkian series as turbidites would come off of the Trenton Shelf and deposit in the slightly deeper water. The most common trilobite in this layer is Ceararis plurhexanthemus, which is a chirurid. The turbidites, or turbidity flows, would disturb the trilobites, prompting them to enroll for protection, and they would then be entombed within the layer in various degrees of enrollment. Walcott discovered these specimens by splitting the layer and noticed that there was calcite within the specimens. But it's difficult to see in the hand samples, so he actually would grind the specimens into thin sections along whichever plane they broke on. So here we have an example of that. This is a transverse thin section of Ceararis through the cephalon, and we can see the exoskeleton, a rim of fibrous calcite all around the edge, and then large sparry calcite crystals that fill in the rest. And this preservation even preserved more delicate structures such as the exopods. And it's really great because it preserved them in 3 dimensions without compaction or displacing them from the body. However, you can see one of the issues with these slides on this slide. Here we have an eye on the right, but not on the left. So these are not actually cut perfectly transverse or axial, and they're often in very odd orientations, which makes it more difficult to interpret the morphology because you're not seeing symmetrical parts of the body. The second most common trilobite in this layer is Flexicalaminii cinerea. And here we have an example of an axial cut. Again, we can see exoskeleton, that rim of fibrous calcite, which completely fills in the smaller voids, and then sparry calcite in the larger spaces. The Museum of Comparative Zoology at Harvard purchased over 250 thin sections from Walcott, and of those, 118 are Ceararis, 65 are Flexicalaminii, 5 are Isotylus, 1 is Mautotanella, and 65 are unidentified. Although the museum has housed these specimens for over 100 years, they haven't been worked on much recently. The aims of my work are to, 1, better understand the taphonomic processes leading to this rare mode of preservation in calcite, and then update specifically the appendicular morphology of Ceararis and Flexicalaminii. And trilobite appendages are quite rare. Although there are 20,000 described species of trilobites, fewer than 40 have preserved appendages, and even fewer of those have appendages that are in 3 dimensions and not compacted. So these are some really great specimens. Brett et al. In 1999 did some lovely work on the taphonomy of the Walcott-Rust, but focused more on other layers, although they did touch on the calcite. So they put forward a mechanism for the calcite preservation, which included that the mud would infiltrate the partially enrolled specimen and needed to cement very rapidly to create a mold of the non-biomineralized tissue, and that this should be in a low iron environment, so that as an explanation for why these were not pyritized. They also then speculated that the partially enrolled trilobite then created a microenvironment which was isolated from the rest of the matrix, and that during decay that would become anaerobic, which leads to an increase in the bicarbonate and sulfide. The bicarbonate can combine with calcium and lead to the localized precipitation of calcite. They also believed that the pyrite, which is sometimes seen on the slides, was replacing calcite and not diagenetically early. So if this is the mechanism, I would expect calcite to be isolated to within the specimen, and that the pyrite would in some way be replicating calcite structures. Here we have a transverse thin section of a Flexicalaminii. You can see the exoskeleton, the fibrous calcite, sparry calcite as usual, but here we have black outlining some of the calcite. So that is potentially the pyrite, but we also have a large calcite vein along the side, which also has potentially the pyrite in it. And it's hard to see if the pyrite is concentrated around the calcite just because of the matrix. So here is an elemental map of iron, and we can see that there is a concentration around those calcite structures, but it's distributed both in the matrix inside the specimen and outside the specimen. The sulfur is more concentrated around the edges of the calcite with less of it in the matrix. So the pyrite does appear to be somewhat concentrated around the calcite. I used scanning electron microscopy to actually look at some of this pyrite and found that it was in rhamboids rather than replicating any sort of calcite structures. And the pyrite rhamboids are associated with sulfate reducing bacteria, so they would actually have been formed very early in decay instead of diagenetically later. And this indicates that maybe since they did not continue to grow and we see pyrite throughout the matrix, there might have been more organic matter in the matrix, so it was not all concentrated at the fossil, so we don't get pyritization. It's important to understand the taphonomic processes before we go on to interpret the morphology. So now I'm going to move on to the morphology of these specimens and some quick trilobite appendage terminology. So they have biramus appendages, which are connected to the body wall through a protopodite, which will always be in green. There can be spines along the ventral edge, which would be called a gnathabase. They have a walking leg, which is called the endopod, which is composed of 7 podomeres, and each podomere can have spines on the ventral side, which would be called endites. Dorsally, the dorsal ramus is the exopod, which is the respiratory or ventilatory structure, and that has lamellae on it. If we look at some of the previous reconstructions of the appendages of Flexicalamini, here is Walcott's in 1884, and in addition to those structures, he had these pink spiral branchi, which he maintained through his 1918 reconstruction, although he converted the exopod basically into a ginkgo leaf and added a gnathabase here on the protopodite. 2 years later, Raymond updated the morphology to get rid of the spiral branchi. They are actually just artifacts from cutting through the lamellae, so it looks like a spiral. And he changed the morphology of the exopod. If we look at Ceararis, Walcott's 1918 version looks very similar to Flexicalamini, with the notable exception of no exopod. Raymond in 1920 also got rid of the spiral branchi for Ceararis and updated the exopod. And then by 1959, authors believed that the endites and the gnathabases were actually preservational artifacts. So here we have a completely smooth appendage and this funky looking exopod that has 6 podomeres and a big club at the end. So we can see that the morphology has been controversial and warrants some restudy. Here is a Ceararis transverse thin section, and you can see those pleural spines along the edges. We can see an exopod up here, which appears to have 3 parts at least, as well as lamellae on it. There are a couple exopods on here, but one is particularly nice, so we'll focus on that one. And we can see podomere boundaries, and along the ventral edge of each podomere, there is a spine. And if these were indeed preservational artifacts, I would expect them to be more randomly distributed and not only coming off of the ventral edge and the distal edge of each podomere. So they're not actually preservational and they are true morphology. So those are the endites. And then if we look where the exopod and the endopod connect, we can see what would be the protopodite, and the spines continue along the ventral edge. So these also have a gnathabase. However, if we compare that same thin section to another transverse Ceararis thin section, we can see one of the challenges of working with these thin sections. So although they're both transverse and the same species, we see very different structures. So here, the exopod looks very club-shaped, which highlights the difficulty and the need to be very careful with where you are in the body and what's part of the structures you're actually looking at when you're interpreting these. To that end, I'm working with Maddie Wascom, an undergraduate in the lab, to use CT scans of partially enrolled trilobites to create, there we go, virtual thin sections that we will then pair with the real thin sections so that we can more accurately interpret the morphology by constraining where we are in the body. So if we look back at the 1959 version of Cearis, I have some updated morphology. Dashed lines indicate areas that will be continuing work, but we can see that there should be endites along the distal edge of each podomere, as well as potentially this dorsal spine. The protopodite should have a naphtha base on it, and the exopod might not have quite so many pieces to it. I will continue this work on the thin sections, but also can now pair it with a large new donation made by Dan Cooper of new specimens. And here we have a new Cearis specimen, and you can see calcite underneath the glabella, as well as in a cephalopod next to it. And what's interesting about this specimen is that it's not just from layer 3, it's actually from layer 8. So the stratigraphic range of this preservation is actually larger than previously reported. And using this new material, of which it's a very large donation, I am searching for new specimens, which I can then take to the synchrotron scanner or serially grind them if that does not resolve the structures, so that I can get 3D reconstructions from a single specimen, because the great thing about Walcott-Rust is that they're three-dimensionally preserved, so it's kind of unfortunate to only get to see a single plane of them as you get with the thin sections. And the other issue with the thin sections is you only get one cut per specimen. So with either of the two methods, we could hopefully get a whole series of the specimens. So just to recap, the pyrite framboids are concentrated around the calcite in the fossil, but also distributed throughout the matrix, especially iron. The calcite preservation occurs in a broader stratigraphic range than previously thought, and calcite also occurs throughout the matrix in veins and blobs. So it's opening the question of like how much of an environmental microenvironment is this, or are they all the same diagenetic events? I will continue this work with more elemental mapping as well as isotopic analysis on both Walcott-Rust specimens and Hertfordshire specimens from the Silurian of England, because that's the only other well-known biota with this type of preservation, although that one occurs in a very different geologic setting, so it will be interesting to compare the two. If we look at the morphology of Cearis, the updates include that the endites are not preservational artifacts. The naphtha bases are present on the protopodite, and the exopod potentially has fewer podomeres. And this work will continue with both work on the thin sections as well as the new specimens, and hopefully working toward 3D reconstructions. I would like to thank both my department and the Museum of Comparative Zoology for funding, as well as the awesome staff at the museum who have been great, especially during COVID. They're super helpful. I would like to thank my lab, especially Maddie Wascom, for her work on this project, and my advisor, Javier.
Great, thank you so much, Sarah. That was excellent. Looks like we have some time for some questions. People want to throw them in the chat or unmute themselves and ask. Yeah, Axel.
In one of the pictures that you have shown from Flexi Calimene, it seems that the pyrite was only on the lower side of these sperricalcite. Is this a common phenomena?
So it's not only on the lower sides, but it's really variable. So you will have a thin section, I didn't actually include it in this one, where you have pyrite really thick around some of it, and then it continues and gets thinner and thinner, but it's not consistent about like which side it's on, although I suppose since these are already prepared, I don't know which way the specimen was originally, if it was upside down or right side up or on its side, but the pyrite is very variable in where it occurs and how thick it is.
Yeah, Joe.
Nice talk. And just wondering, is, of course, Herefordshire, virtually everything is preserved in the same manner. Is it, from the new collections, as far as you can tell, still just the trilobite soft tissue? Is it being preserved this way? Or is there anything else with soft tissue as well?
So that's all I've seen. So like the cephalopod is that not non-biomineralized, it's just now filled with calcite. The trilobites are the only one with the non-biomineralized tissue. I think I've seen crinoids, which are also filled in with calcite. So I haven't seen any other species or like non-trilobites with non-biomineralized preservation like this.
Interesting. Oh, well, keep an eye open.
Yeah. Unfortunately, you have to destroy the specimens to check. But with the new material, the large donation, I have to destroy it all anyway, so.
I guess I'll ask a question. Do you have a chance or opportunity to look at any of these with the CL if they're uncovered?
I am trying. There is not a working CL around, but Tom Hegna actually has looked at them with a CL detector, and he saw that there is a bit of a shift between the fibrous calcite and the sparry calcite.
Sure, so that might help. That might help with your paragenesis story.
Yes, I just need to locate one.
Yeah, well, come on down to Boone. I'll open the door for you here.
Perfect.
Have you even compared how the pyrite looks like in the matrix compared to inside the trilobites? I wonder if those would have a similar.
Not yet, but I would be curious to see.
Yeah. All right. If there aren't any other questions, I guess we can move along. So thank you, Sarah. Great talk. So next up, we have Ciara. Am I pronouncing your name right?
Almost, it's Ciara.
Ciara, okay, thank you.
Yeah.
So Ciara is here, and she will give a talk with her co-author, Alycia Stigall, entitled, How Do Ecological Niches Evolve During Ordovician Environmental Change? A Test Using Laurentian Brachiopods. So hopefully you can share screen and throw your slides up.
Yep.
Ah.
Hold on. Okay.
A window into the past. There we go. All right. Thank you, and take it away.
Okay. So thank you guys for coming to my talk. So I'm excited to talk with you today about my master's thesis research. For my project, I am investigating whether and how brachiopod genera altered their ecological niches when faced with substantial abiotic and biotic change during the Ordovician period. A species' ecological niche represents a fundamental relationship between that species and its environment, and because the niche is integral to species ecology, by quantifying it or defining it, we can begin to understand how changing environments can dictate species response. So the purpose of my project is to investigate how species respond ecologically to different types of environmental change, specifically by looking at changes in niche occupation. Some of the questions I hope to address are whether or not niche occupation does change throughout the Ordovician period, and if it does, what the trends in those changes are in relation to different environmental shifts. I also want to investigate how generalists and specialists respond to the same environmental pressures. These questions should help inform our understanding of modern niche concepts and help us to understand how Ordovician changing environments affected the ecosystems of that time. So the niche and niche concepts is defined in different ways throughout the ecological literature, but for my project, a niche is a environmental space. In other words, a niche is just defined by the necessary environmental characteristics that allow a population to persist in a given area. So the real-world geographic distribution of a species represents what is known as the occupied niche of that species. The occupied niche can be formed, or is formed, as it is conceptualized in this BAM diagram here, by a combination of the necessary abiotic environmental factors, non-exclusionary biotic interactions, and viable dispersal or movement capabilities of the taxa, which we can see represented by these 3 circles. The occupied niche is the niche that I hope to quantify in my research. So the Ordovician period is a particularly good time to investigate how changing environments impact species niches because it's a time of profound environmental change, both in the abiotic and biotic dimensions. Abiotic changes include a cooling of the climate towards more modern levels and culminating in Hernanchian glaciation at the end of the interval. Sea levels also fluctuated throughout the period, and more localized changes like the tectonic orogeny affected local areas. The marine ecosystem itself also changed in substantial ways throughout the Ordovician. This includes the goby, which is the rapid diversification at family and genus levels. The ecosystem structure also became more complex throughout the Ordovician, such as through the development of bioturbation lifestyles and epibiont evolution, creating more modern ecosystem structures. And then finally, we have a lot of more localized biotic interchange events like the Richmondian invasion, which would place competitive pressure on both local and invasive marine taxas. So abiotic and biotic environmental shifts like these are still occurring in our modern world, impacting species and their ecospaces. And the degree to which an environmental change will impact a species depends a lot on how much influence that environmental condition has on the species' ecological niche and the rate at which the change occurs. For modern conservation efforts, it's really important to understand how species will respond in the long term to changing environments. And one of the best ways that we have to try to understand how they're going to respond is by looking at the ecological niche and trying to predict species response to specific changes in the environment. One of the classic ways that we can use to try to quantify a species' ecological niche is through a method known as ecological niche modeling, or E&M. Ecological niche models are quantified representations of a species' occupied niche, and they are produced by correlating the geographic distribution of that species with the abiotic environmental characteristics in that same time and place. So this geographic information is correlated into an ecological space using a modeling algorithm to create a niche model, which is what this is representative of here. Niche models can continue to be conceptualized in this hyperdimensional ecological space, or they can be projected back into geographic space. And the versatility of how we can conceptualize these models makes them really useful for answering a variety of ecological questions. So E&Ms can be used to create species distribution models, to compare niche breadth between related taxa, to estimate the invasion potential of different species into new areas, and potentially to predict the response of species to changing climate or environmental scenarios. So as I said before, quantifying the species' ecological niche using E&Ms is particularly useful for modern conservation efforts because we can try to see how our own changing climate will impact those species in the long run. So this is just an example of a paper that used E&Ms to try to predict population dynamics and species response using the niche model for the particular species they were looking at. However, modern applications of E&Ms rely on either very recent or current occurrence and environmental data, and they can't usually compare models through time. So they have to really assume that the niche of a species will remain consistent through time. If a species was instead able to change its ecological niche in response to new environmental conditions, then the predictive power of that niche model would be significantly decreased. So in my project, I'm hoping to create a series of models using fossil data that can be compared through time in order to see if niches do remain stable in response to different environmental changes, and if they evolve, what the relationship between those two are. So in order to look at this question, I need both occurrence and environmental data. For my occurrence data, I am using brachiopod fossils found in exposed Ordovician strata, specifically focusing on the Laurentian paleocontinent. I downloaded my total data set from the Paleobiology and IGCP databases, and in order to ensure data quality, I went through my data set, sorted out missing information, flagged any possible errors, and verified the taxonomic consistency. This is just a map showing my final data set that I have now, and I currently have 30,344 occurrence points total. So once I had my occurrence points compiled, sorted, and cleaned, I was able to separate them into time bins no greater than stage level. I then selected genera to use for modeling based on whether or not they had at least 10 occurrences per time bin for at least 2 consecutive time bins. And this is just an example of 3 genera that I've plotted into the first 3 stage slices of the Katian. I also restricted the occurrence points of my selected genera to areas that had good environmental data coverage. So that's what these black or dark gray boxes represent in these maps. And we can see in this first map, I have Hebertella genus occurrences plotted within this specific geographic region. And then I summarized these original occurrence points into a single point per 1 degree grid cell. And this is because my environmental data is very general. It's in a raster file format at 1 degree grid cell size. And including a higher resolution of occurrence points would only bias my final niche models towards areas that have a higher data collection rate. So I've just taken these, I think there's like 10 points here, a couple of them overlap a lot, and summarize them into 4 points. So in addition to occurrence data, I need environmental data. For modern E&Ms, environmental layers have already been produced for this purpose, but because I'm looking at fossils, I had to collate my own using the sedimentary record. The 6 characteristics that I've been using include limestone type and percent, bedding style and thickness, percent mudstone, bioturbation, percent siliciclastics, and siliciclastic structures. These characteristics were chosen because they are believed to represent environmental conditions during sedimentation, like water depth and energy level, oxygen level, substrate type, things like that. And this stratigraphic column is just an example of how sedimentary information is summarized across an entire outcrop into single values, such as like percent clay, that can then be plotted geographically. So once I had data points for my environmental layers, I then plotted them in ArcGIS Pro. And we can see here just an example of water depth or inferred water depth, which is based on several different stratigraphic characteristics. Darker points representing deep water and lighter points representing more shallow water. We can see that these points cluster in these distinct regions, which represent areas of very good Ordovician outcrop that have been well detailed in the sedimentary literature. Using the ordinary kriging method, I then interpolated a continuous surface of inferred water depth across both areas. We can see once again that darker areas represent deeper water, lighter represents more shallow water. However, these interpolations are pretty preliminary right now. I'm currently working with some issues related to the scarcity of my data, which is pretty clear when you look at this example in particular. To solve this, I'm trying to collect more environmental data points that I can use to create a more extensive data set. And also I'm looking into different interpolation methods that might be more applicable to my data set itself. Either way, once I created my environmental layers, I then converted them to a 1 degree raster grid format in ArcGIS Pro, and then brought them into R, where I was able to stack them. Stacking required that they all line up exactly, and areas outside of my study regions are given a no data value so that they won't be influencing my models. I was then able to bring in my preferred genera and combine them with these environmental layers in each time slice in order to create a niche model using the Maxent algorithm. So Maxent is the current standard for E&M analysis. And it works particularly well with my data because it works with presence-only data, small sample sizes, and limited environmental variables. This is just an example of how Maxent works by taking occurrence points represented by these orange crosses and different environmental layers and correlating them to create a maximum entropy distribution of the species fitness based on the limitations imposed by the environmental layers. So we can see in this example, this is a species distribution, or fitness distribution for this species that has been projected back into geographic space with warm colors representing a high species fitness and cool colors representing a low species fitness. This model down here is just an example of how this niche model could be conceptualized in a multi-dimensional environmental or ecological space, where the ellipse represents the model itself and it's encompassing the occurrence points of the taxa. So in R, in order to create the most optimal model for each time slice, I first created a series of test models using the E&M eval package. And this compared various different beta multipliers and feature classes used by the algorithm to try to find the best parameters to represent that particular genus in that time slice. So out of this test set, I then selected the model parameters which produce the highest AUC value and lowest emission rate. And I used those parameters to create a final model using the Maxent package that I'll show you a few more of in just a minute. We can see that in this particular example for Hebertella, that the occurrence points all do plot in areas of high species fitness. Once again, that's represented by warmer colors, and they are not plotted in areas with the low species fitness. So this is an example of the Hebertella genus models plotted through time for the first 3 stage slices of the Katian. We can see that there is a change in the distribution of high species fitness between the Ka1 and Ka3 stage slices. But unfortunately, several of the models that I've created so far have been not very good. They've been sort of inconclusive, in which we can see an example of here with the Ka2 stage slice. This model has an AUC value of only 0.5, so it can't be really differentiated from random. This could be because of my the poor resolution on my interpolated environmental layers, or the small sample size I'm using, or the fact that I haven't used Maxent very much. But either way, as I move forward and finalize my data set and get more familiar with Maxent, hopefully I'll be able to create much more robust models. So I'm currently in the beginning of my second year of my 2-year master's. So over the next several months, I will be completing my environmental data set, finalizing my environmental layers, and optimizing my model creation methods, so that in the spring, I'll be able to present a much more complete set of models that I will have drawn conclusions about niche stability patterns from. So there are basically 2 n-member degrees of niche stability which I might see in my data. Either the niches will be identical through time, indicating total niche stability, which we can see an example in this diagram here, or the niches are completely dissimilar, which would suggest niche evolution. So niche evolution can occur through different forms of species response. The first would be if the species expands its ecological niche to encompass a more broad set of environmental parameters, which we can see in the second column. The second would be if the species contracted to a subset of its former preferred niche. And finally, the species could shift to an entirely new set of environmental conditions as it adapts. Previous similar studies to this have suggested that different types and rates of environmental change are related with different patterns in niche evolution. So a previous study found that the gradual sea level change was more closely correlated with niche stability, whereas rapid biotic invasion events were more closely correlated with niche evolution. Generalists and specialists will also be compared in order to see if they respond differently during the same intervals. And it is typically believed that generalists have a much broader ecological niche than specialists, so theoretically they could expand or contract their niche more easily than specialists can. I hypothesize that the different types and rates of environmental change will correlate with different patterns in niche stability and niche evolution. So in conclusion, the Ordovician period changed significantly in both the abiotic and biotic dimensions, impacting evolution and ecology. And by applying niche concepts to fossil data, I will be able to determine the degree to which different types of environmental change impacted species niches and whether those niches remain stable through time. This will help inform the validity of modern E&M applications and help us to understand long-term ecological patterns. Thank you.
Thank you so much. That was a great talk. We have a couple minutes here if people want to pose some questions.
May I ask a question?
Yeah, please.
I'm a bit curious about your data resources. You mentioned two databases, and one is PBDB. Everybody familiar with it, but the other one seems a bit. We are not so familiar with this one. So, can you explain? Maybe give a brief introduction about this database.
Oh, which one did you mean?
The
iDigBio.
DigBio.
Something.
Yeah.
Yeah.
So that one's pretty similar to the paleobiology database. It's in a different format. I haven't explored a lot of it because I mostly focused on just downloading the brachiopod data, but it definitely didn't. It took a lot of effort to try to fit them both together because they weren't in the same format, but it included a lot of the same information. Like obviously taxonomic names and locations and collection dates and all that. I don't know if that answered your question.
If I can jump in?
Yeah.
The iDigBio database is based on museum collections.
Thank you.
And it is museum specimens and the collections there. And it's much, much bigger than the paleobiology database. And there was a recent paper showing that actually there's a lot, like 10 times as much data in museum collections as there is in things like the paleobiology database. And if I can ask a question.
Thank you.
Yeah. You did the ordinary cridging.
Mm-hmm.
There is in cridging the maps is also a way to determine the uncertainty.
Yes.
What did your uncertainty on your maps look like?
So obviously for areas like where the points were quite well clustered, the uncertainty was relatively low, especially depending on some of my environmental variables seem to correlate really well spatially. Other ones showed very poor spatial correlation,
right,
which was part of the problem. But in general, the uncertainty varied a lot. And in a lot of areas, it was pretty significant, which is one of the reasons why I was like, I need to find a way to fix that if I can. But yeah.
Now, again, you run into the risk of overfitting the model to the data.
That's true, yeah. Yeah, I definitely need to explore it more.
There's a question in the chat from Lena. She's asking, do you have a final total of taxa that you'll include in the final analyses to meet these criteria?
No, I'm gonna do as many as I can. Currently, because I've spent so much time focusing on just cleaning the data sets and compiling my environmental data, I've only done like 5 of my genera. Hopefully I'll have more than that so that I can actually compare quite a few different genera, but I will at least have, like, I expect 8 or at least 10, somewhere in there. Hopefully a lot more than that. We'll see how it goes though.
Okay. There are no more questions. It looks like people are throwing some links in the chat to these different databases, so feel free to check those out. But in the interest of keeping on time, we'll transition to the next talk. So thank you, Ceara. And our next speaker is Alejandro Corrales-Garcia, Jorge Esteve, and Mateo López-Pachón. Is Alejandro here? I think I saw the name. All right, great.
Do you hear me well?
Yep.
Okay.
If you would share your screen, and as he's doing that, I will introduce the talk. So Alejandro's talk is entitled Burrowing Assessment of Ilianus sargi janison and Megaestapsis exitenata from the Middle Ordovician of Sweden.
Yes, right. The names are very difficult.
Certainly from this angle. I'll let you correct me here. So take it away, please. Thanks.
Okay, thank you. In this story, this story is about the burrowing assessment of two trilobites from the Middle Ordovician of Sweden. And it's actually a continuation of a part of my thesis for my degree in geosciences at the University of Los Andes. So, as many of us know, one of the most important events in evolutionary history of trilobites was the Great Ordovician Biodiversification Event, the GOBE, that substantially increased the taxonomic, morphological, and ecological diversity of various groups of trilobites. During this time, as you can see here, there was a change of from a general Cambrian morphology towards a greater variety of morphological novelties. A good example of this is the variety of cephalic forms, which, as we can see here in the from the studies of food, they achieve a maximum in this event. These changes will occur in a convergent way between different groups of trilobites, thus allowing them to occupy different levels within the water column and therefore live different lifestyles. As we know, studies in paleoecology could become a little tricky, and especially in the cases of extinct clades such as trilobites. And sometimes evidence is strong, is specific, and is very useful, like these cases I'm just showing you. However, by looking into exceptional cases, one could be overlooking ecological relations and behaviors that fossil record just apparently misses. And when we came across with structures, we're looking morphologies and evolutionary trends, we cannot help but wonder if they have a purpose, or if they confer an advantage to the individuals, or if they were just a byproduct of evolution. In this case, a good amount of trilobites came up with enlarged cephalic structures, or cephalic snouts, like I would call them, and in various ways. 4 hypotheses have been laid in the literature until now to explain this kind of processes. One, a passive protection function against predators, causing a mouthful effect. Second one, to prevent burrowing into the sediment, causing a snowshoe effect. And on the contrary, as a burrowing tool, allowing them to dig well. And 4, a sexual selection, and this is very common when morphology, like when amazing structures came up, sexual selection will always be an hypothesis. So what do I compare a sword-shaped front of a trilobite? The best is to look into another shape that is classic for the kind of behavior one is trying to test. And the answer was infaunal bulldozers, bulldozing animals, many of them with blunt and wide shaped heads, a characteristic that many trilobites had, and a real tangible trend in also other burrowing animals like moles and snakes that are vertebrates. So, taking into account that the anterior part of fossil organisms should be more resistant to pressure than non-fossorial organisms, our objective is to use 2 trilobites from the Middle Ordovician, Elinus archi and Megistaspis extenuata, to quantify the difference between the two infunctions of the 2 different ecomorphs, especially as the shape of the exoskeleton allows them to better adapt to fossorial forms of life. In order to quantify the forces responses of our models, we use finite element analysis, a tool that is the kind of the ultimate tool for simulating processes applied to any bidimensional, 3-dimensional surface. There are a lot of examples of this methodology in biomedicine and in vertebrate fossils, especially the studies of Rayfield. But for invertebrates, there are just a few in the literature. For trilobites, there is just only one example on the where to be published by Esteve. Which is weird because we know trilobites are a very large clade. So maybe it's about time to put this kind of analysis in these organisms. So we use a combination of softwares to train 3D models reconstructed from very well preserved specimens like Blender, MeshLab, and Fusion 360. And the final results are measures that we can use to simulate forces. But for this investigation, we just used the cephalon, as it is the part that undertakes the first contact with the soil, and also becomes easier for the computer because this kind of analysis require high computer power. We put a wide range of forces applied at a reduced area at the anterior margin of the cranidium, and the measures were fixed in a ring of elements in the posterior part. Can you see my pointer? I don't know. I think it not. Okay. And in the same way that that Stevie and collaborators use in the work on the review. So we obtain maximum deformation, maximum stress and strain for each of the models. And as we can see here, it's very evident that Megistaspis extenuata constantly presents higher values of the 3 components measured in all the applied forces. This, for the rest of the presentation, I'm going to show you just the results for both models, for both species at 5 Newton, because they were all very similar in aspect, just for visualization. So for deformation, you can see here not just that the higher values of deformation are very notorious in Megistaspis extenuata, but also the distribution patterns of both models are very clearly represented in a kind of banded way. And one may think is the position of the maximum deformation in Ilano-Sarsi, located precisely in the place where the load was applied, whereas in Megistaspis extenuata, it occurs at the middle of the enlarged snout, causing this kind of bending. Another remarkable point is that the genal spines and the lateral border also presents higher deformation values. Now, if we see the direction of the deformation, is pretty remarkable, the difference between the 2 models. Posterior and outwards directions for Sarsi, and an almost exclusively vertical vector for Megistaspis extenuata. On the other hand, we can see a similar distribution of values between stress and strain. High values in the front of the glabella and in the limits with the postocular and palpebral area. And additionally, there are high values in the place where the load was applied, and for Megistaspis extenuata, also along the axis of the snout. However, the maximum stresses and strain were always located at the edge of the occipital rings in the posterior part of the cephalon. So, what we can obtain from this data? First of all, that the direction of the deformation of the cephalon could be playing a major role in the fossorial trilobites. This is the more prominent difference between the 2 models. And on the other hand, it seems that the rest of the skeleton was pretty important for the fossorial habits. The connection between trunk and cephalon presented the higher values in stress and strain. And the mechanic of the connecting tissues needs in a future, hopefully close future, to be integrated as well. And given the higher values of Megistaspis extenuata, we can think that the enlarged snout of trilobites was not as good for burrowing as having a blunt surface. Therefore, it was probably not suited for digging deep burrows. So what was that for? Maybe an answer to these and other open questions that I laid out in the presentation, it would be good to simulate by other approaches like computational fluid dynamics. The different layers of soil to simulate in the different layers of soil and in the water column by mimicking the physical conditions of the infannual and benthic habitats. So to sum up, there is a generalized trend in the response in the trilobite cephalon to different loads, like high values in the glabular area, for example. The models obtained in the study for Ilinosarsi and Megistaspis extenuata allow us to see the mechanical reactions for a complex three-dimensional trilobite cephalon for the first time. And that may be the most important one, fossorial habits is plausible for Ilinosarsi, which shows numerically a marked resistance to the imposed forces. And in contrast, Megistaspis extenuata was probably not suited for burrowing, and it is outlined as maybe a benthic organism. Finally, I want to say thanks to Mateo Lopez and Carlos Rodriguez for teaching me the difficulties of these methodologies. For all the people in the lab at the Los Andes University for aiding me and allowing me to work with them. And especially grateful with Jorge Esteve for guiding me in this investigation, these and other investigations actually, and for introducing me into the fantastic world of trilobites. Thanks.
Great. Thanks so much, Alejandro. Great talk. So we have some time for some questions. There's Roy posed a question in the chat. Roy, do you want to ask your question or?
Sure. Why not use horseshoe crabs as a model? They are classic arthropod burrowers. There are also some crabs that burrow. Nico Savazzi has worked on those.
Yes, I just use trilobites because I forgot to talk about it, because those models, those species were coetaneans. They lived in the same area at that time, so they're probably in the same environment. And
I'm using this as in the beginning. You mentioned using vertebrates as a representative of animals that burrow.
Ah, okay. No,
there are modern animals that burrow.
Yes. You're actually right. Yes, maybe I should put a photo of a horseshoe crab too.
And all second question, these are calcitic skeletons. Do you have anything about what the strength of these would have been?
Yes, in the finite element analysis, as it is computational, you have to put all the physical properties of the material you're working with. In the studies of Emily Rayfield, they used a bovine bone physical attributes, but for this work, I used the normal calcite attributes. Yes, as it is the one that is in the trilobite exoskeleton.
Great. Sophia, your question?
Hi, can you hear me?
Yep.
Yes.
Yes. Hi.
Thank you for your presentation. My question is also related to the horseshoe crabs, because in your model, you're assuming that burrowing capacity is related to the morphology of the cephalon. But for example, the ecology studies on limulas, on horseshoe crabs, they show that burrowing involved both leg motion and body flexing, but not necessarily the cephalon morphology. And in horseshoe crabs, that cephalic morphology is more related to the hydrodynamic lift of the body, but not related to the burrowing.
Okay, that's a very good question. Well, the reason I laid out this, I'm gonna get back. These two animals were, first of all, Inanusarsi is proposed because it's probable that the legs didn't like reach beyond the cephalon. It's just too wide. And this occurs too in that kind of mold that actually chews the earth around, and for snakes and amphibians that have no legs. So I'm just comparing the cephalon in this time. But it will be very good to have.
I understand, but I think that horseshoe crabs are so much more closely related to trilobites that probably the mechanism will be much similar to trilobites than, for example, these vertebrates. I think so. Well.
Yes, I will have to revisit that. Thank you.
I think we have time for another question or two. All right, I guess you've left us all satisfied. So thank you so much, Alejandro.
Okay, thank you.
Yeah, and so I guess we'll move on to our final presentation of the early session, not so much the morning session for us all. So next up, we have our keynote talk for the day. We have Jorge Esteve and Mateo López-Pachón. So I think, yep, it looks like Jorge's here. Great. So Jorge's talk is entitled Swimming and Enrollment in a Mesopelagic Trilobite, a New Ecomorphological Advantages in the Middle Ordovician Ocean. All right, so we can see your screen, Jorge, but I don't know if your audio is enabled yet.
Yes, now I'm talking alone.
Okay.
Sorry.
Right.
Okay, thank you very much for the invitation, I was saying. Yeah, first of all, I would like to say that, well, today I'm going to talk about this little trilobite from the Middle Ordovician of Czech Republic. And most of my career has been focused on functional morphology. Enrollment is one of my favorite things. And when I found this enrolled trilobite, it was a big surprise because, well, this is a classic pelagic trilobite. And I thought, oh, why this guy need to enroll? So, well, I tried to explore in this talk this question, but also other things. This is working or yes. So the stand timing, duration, and magnitude of diversification and extinction events are in constant debate. And this is, from my point of view, this is great and really necessary to understand the biodiversification dynamics, and as well as the duration and magnitude of the radiation and extinction events. So, well, I'm not going to focus on this, but I think this is important, okay? So the Cameron expulsion, Great Ordovician biodiversification event, and the Mesozoic marine revolution were established all the marine diversity and ecology pretty much fast in geological terms. In this talk, of course, I'm going to focus on Ordovician, but also worth to mention that we have to pay attention on the Cambrian explosion, of course, and all the Cambrian times, because, well, as you will see, we know pretty much nothing about what's going on the water column. We know a lot of things about the benthos and the benthonic animals, but almost nothing about the water column, okay? So these diversification gradations can be analyzed from two different perspectives. The benthic in faunal transition, where we can find two important phases, the early phase during the Precambrian Cambrian until the Cambrian series 3, and the second important phase, which happened during the lower Ordovician until the Middle Ordovician, where there is a huge increment in the number of bioturbation structures, as well as the depth of these structures and the morphology, etc. So these infaunal habitats were colonized with a structure which are getting more and more complicated and more complex and deeper. And this is very well known with a number of publications dealing with such topic. But as I say, what's going on with the water column? Now, well, we have a very good idea about biodiversification dynamics during the Cambrian and Ordovician, but this sort of biodiversification analysis includes pretty much all taxa. Anyway, we can have a look at this data and see what's happening, okay? So although lately new studies compile invertebrates but by periods of time separates on how different styles of life, here in this slide, Stigall and others include within the nectobentonic animals all trilobites, for instance, here. So for me, this is, well, I guess Jonathan Adrien and in these papers, they mentioned that this is a nectobentonic animal. And well, for me, it's kind of weird. And actually, we don't know if these trilobites or all of them were nectobentonic. And now I will explain why. But this is okay. It's no big deal. This kind of studies help us to understand the biodiversification fluctuations. And later we can explore more to see what happens behind these curves. I think these curves are very important. And as I say, these sort of plots are in constant modification and discussion. So it's not a problem from my point of view. In this paper also, Stigall and colleagues also separate between benthic and planktonic, including among the pelagic predators only cephalopods, which is, I guess, the only data that they had in that moment, the only available data. And anyhow, these sort of studies show us that our knowledge about nectonic diversification is poor, and especially in the early stages of the animal evolution. So we have to explore a little bit more about the nectonic diversification. There are some words, but not too much. In 2010, Krug and others documented for the first time a major radiation of diversification. They call the Devonian Age of Species. And for these authors, the Devonian necton revolution was a sudden revolution of the necton, accounted for more than 85% of the non-bentonic marine diversification by the Carboniferous, compared to less than 15% in the late Silurian. So this is a huge amount, and seems like Devonian was really important. So in addition, the Devonian necton revolution coincides with the increase of the benthic predation resistant morphology, and this was the trigger for the Mesozoic marine revolution after the Paleozoic, of course. So although nectons are an important component of the global biosphere, an important contribution to the trophic complicity and nutrient cycling, this work published in 2010 was pretty much the first to deal about this, from my point of view, very important topic. After this, we have this also very nice paper published by Wellen and Briggs in 2018. And they were using new data and analyzed patterns of nectonalization during the Paleozoic, and they expanded all the taxonomical coverage and made a new sample standardization and increased the ecological resolution. And in this work, they found they found that the nekton and neonectonic were well established not in the Devonian, but previously to the Devonian. So they found that this diversification in the Devonian was not really so fast. It was something more gradual, more that it was coming from the Cambrian, especially in Ordovician, and I think we have to pay attention in the Ordovician, and not only in the Cambrian. So this is true, and of course, during the Devonian, we can find a relatively increase of the nectonic faunas, but although data shows in Wallen and bricks a little decrease in the number, so Devonian is important, but we have to look at these other times. And thus, Wallen and bricks suggested that the Metazoan colonization of the water column was, as I say, more complex and more gradual than previously was understood. So in this work today, we want to increase our understanding of the ecology of pelagic or mesopelagic trilobites and try to figure out the transition between benthic and pelagic habitats in trilobites. So the transition was not that sudden. So we want to see how occur in this very interesting group. So we can divide the nekton into two different groups: nectobenthic animals, those swimming close to the sea floor, and eunectonic animals, so those which are not swimming near to the sea floor. And everything is okay? I don't know. Yeah, I guess so. So, well, for Forte in 1985, he showed the first analysis of fluid dynamics in a couple of trilobites, and for him, those very well streamlined trilobites were the best adapter for swimming, of course, so no problem. But he didn't really talk about any transition or how began swimming in trilobites. So, well, we can say that these well streamlined trilobites are eunectonic, while these not so well streamlined trilobites were, well, nectobenthic. Nectobentonic, well, maybe we will see. So the other important work about this topic was by Sino and others in 2014, and they assessed the shape of some interesting forms, Ordovician forms, for swimming. So they test the different the distance, how the distance of Ipodicranitus affect to the hydrodynamics of this trilobite. So the distance between the animal and the sea floor. So different distance, and they found very interesting results. So they found that thanks to the large hypostome of this interesting trilobite, they allowed a very effective way to swim very close to the seafloor. This is a very interesting thing. And we can see that this is a real nectobenthic animal. Why I say this? Because, well, in this case, in this calimenid, well, we cannot say that this is a nectobentonic animal. This is it's a bentonic animal, and that's my problem with that course in the paper of Stigall last year. So they include all these animals within nectobentonic when they are not really nectobentonic. They are only bentonic. So that's a big difference between, for me, this is a big difference between these different forms, okay? So, well, in this line, we are right now working, trying to understand how was the beginning of swimming, how was this transition. And it seems clear that the evolution was not too fast as Krog and others proposed it a few years ago. So maybe we have to look at the trilobites with new eyes, new perspective. So, but how to assess nectobentonic life habit? This is not an easy task. This is not something that we can address very simple. So now we are addressing this problem by means of computational fluid dynamics simulations in different trilobites. And I would like to show you our first step to understand this transition, just to see how began the transition between a real benthic animal and a nectobentonic animal. So we assess the hydrodynamic of Placoparia. This is the first trilobite that we assessed 1 year ago or so. And this is a nice trilobite with unique novelties to achieve a very novel enrollment style during the Middle Ordovician, very important for the ecology as well. And it was our first model to understand whether nectobentonic life habit was common or not among trilobites, or how was this transition between bentonic animals and nectobentonic lifestyle. So, well, we, as I say, we carry out some computational fluid dynamic simulations, and the results are super nice and very cool results. And we can see that when Placoparia was close to the seafloor, was very unstable, was very, very unstable. But when this is close to the seafloor, and this is 12 millimeters from the seafloor. So we have all this turbulence, all this movement, and that means that the stability was not really good. So we can see some turbulence also here, and we don't have any turbulence when they, well, just a little bit with a slow velocity, but we have pretty much nothing with when the animal is just a little bit above the 12 millimeters above the seafloor. Okay, so well, this is mainly because well, first of all, we cannot see that this animal was a real swimmer, because the drag coefficient, this is the drag coefficients, are not so good. So this is not a real swimming, but just I want to explain why we believe it's important to study this animal. So this instability, this is because the positive leaf will cause an animal to be dislocated while negative leaf will help to the animal to maintain contact with the substratum. So we have this positive leaf here, very positive leaf. So this very high positive leaf is what is doing this animal to be not very stable close to the substratum, close to the seafloor. But when we have this negative leaf, so you can see 12 millimeters from the seafloor, the leaf coefficient decrease. So it's easy to the animal to keep stable. So I don't want to say that this animal could swim. As I say, the drag coefficient is too high, but probably this animal was able to do some kind of hopping locomotion, something like that. So, well, maybe this was the beginning of swimming, so something like this. So probably swimming didn't start just like we are going to see now with Placoparia, but something more progressive. So this animal is a nectobentonic. Well, in this case, I don't think so. I think this is a bentonic animal, but with some kind of hopping locomotion thanks to this balance between leaf coefficient when the animal is close to the seafloor and a little bit above the seafloor. Okay, but how is a nectonic animal, a real swimming animal? Well, it has a general outline is elliptical and lacking projections, so it's streamlined. We can see that the snout or nose is very small in this case, but other cases is very elongated. Okay, very efficient to reduce the drag. And also we can see that the eyes in the case of trilobites, they are tucked in the side of the head, so the eyes don't interrupt the general profile of the trilobite, as we can see here. So eyes are very well in a very well position to keep a good hydrodynamics. So according to Forte in 1985, Microparia fulfilled the features to be a good swimmer. But Richard Forte also put other criteria in this. He wrote that also we need to see what the environment and the geographical distribution. So we wanted to see what is the geographical distribution of this trilobite. So according to the PDBDB, the Cyclopygi have a widely distribution in North America, South America, Europe, also in Asia and China and Australia. However, Microparia has not that widely, they are not so widely distributed. So they are in deep water sites. Also, it's important to mention that other Cyclopygids, they are in many different sites, deep water and shallow water. But in the case of Microparia, it's found only a little bit in North America, in Czech Republic. The specimens today are coming from Czech Republic, England, and just in few sites in Asia. And we don't have this genus in Australia. So on the other hand, as I began this talk, this specimen was found in the National Museum in Prague in this very nice position in enrolled. So enrollment is an adaptive behavior, very common among benthic trilobites. Why benthic trilobites? Well, protection against predators and against Bruton deposits, against storms and things like that. But this is the question, which was the purpose of this behavior of a pelagic trilobite? How was the hydrodynamic of this trilobite? How worked within the water column? So these questions are those that we want to address and we want to give an answer in this talk. Something is, yeah. So we have the fossils, and my apologies, I have good pictures and good themes, but because this coronavirus theme, my good pictures are in my hard disk in my office, and I couldn't go to my office. And that's the reason, because, well, we did a CT scan in the Prague Museum in Prague, in the National Museum, but this is not, my apologies, this is not the CT scan of this trilobite. As I say, I couldn't go to my office, so I'm so sorry. I couldn't imagine this in March, like probably the rest of you guys. Well, but with the micro CT scan, we can build these reconstructions using this software. We have these reconstructions in the office. And finally, we can create these models. I have the model. Otherwise, I cannot show you the talk, but I have the 3D model in Blender. So with this, we can create a mesh, and there were different simplifications in the Microparia model in terms of geometry. So only we use half of the mesh for Microparia. And yeah. Okay, yeah, so since Microparia model is symmetric, we can use only half of the mesh just to save computational work, and it's easy to move all the meshes and all these things. Of course, I removed the legs. We don't have any idea about the legs of the pelagic trilobites. We have no idea, no clue about how were the legs. So I'm very happy if some of you can tell me a clue about how was the legs of these animals. So we didn't include the legs, but again, if we include the legs, the analysis of the computational fluid dynamics could be very difficult to carry out and very, very, well, almost so far, almost impossible. Well, we did 8 meshes convergences that were performed for each domain to establish the dependence of the results, and we used the drag coefficient as parameters to determine the convergence. The different configurations were performed so to understand the hydrodynamics behavior of this specimen. So we use the reconstruction in a prone position and also in enroll position with different flow directions. Front, rear, cross 45 degrees from flow, cross flow, 45 cross flow from the front. So these are the experiments that we did. I'm not going to show you all of them. I'm going to show you only the most important. For the boundary and initial condition, the working fluid was ocean water using the properties of the ocean sea that we obtained in a number of publications, and we consider isometrical, incomprehensive, and Newtonian the water, okay? So the microbarrier position in all the models is far from the sea floor, so probably, well, the sea floor is a very important thing, so the hydrodynamics is going to change dependently if we are very close to the sea floor or very far. So for these models, we put the sea floor very far just to avoid any interaction between sea floor and the 3D model. So different inlet velocities were tested, as you can see in this table, for prone position and enroll position in microbarrier. So I'm going to show you just some results about the drag coefficient with from flow. So the drag coefficient in the fluid dynamics is very low, which is not, this is not weird, since the animal is very well streamlined. So the results show that the velocity decreased rapidly where it first encounters with the microbarrier reconstruction, with this the steep velocity gradient. So the boundary layer is developing as the flow approach, and the lower and upper in the lower and upper margins of the microbarrier model. So the field velocities shows a vortex. You can see this vortex here, and the body of microbarrier is very elongate, and the vortex of the microbarrier is very elongate, just below the body. On the other hand, we have a wake, very symmetrical wake, which is here with the frontal flow, and the wake is approaching, and the vortex is approaching with high velocities, which is pretty much something normal, as is pointed with these white arrows. When the animal is enrolled, we have also very interesting things. The wake shows two vortices, you can see here, one vortex here, one vortex here, and in all the enrolled simulations, so we have a very asymmetrical wake. And this is interesting because with, especially with high velocity, because this is making to the trilobite to push up. So we increase, so this is drag coefficient, but we are increasing the lift. But we will see later, this is very well balanced by the trilobite, because the trilobite has a negative pressure in the pygidium just here. So we have this wake that pushes up and a negative pressure that pushes down. And this balance is very interesting for this trilobite. With the drag coefficient, when it's arriving from the rear flow, also we have a very interesting results. And also we have a wakes very close develop and closer to the trilobite body in higher velocities, and providing also positive lifts just here, okay? So worth to say that the vortex takes particles directly to the ventral side of the head, which is pointed out just in this blue arrow. So here you are not going to see very well, but in this other reconstruction, you will see how the particles are coming just directly to the mouth. This is a ventral side. So we interpret this like a passive feeding as assisted by the flow. So the animal just was floating and particles move directly in the flow directly to the mouth of microbarrier. So we can see this in different views, And when the animal is swimming counter current, the vortex takes also particles to the mouth of trilobite, but it's more evident when microparia is oriented in the same way of the flow current, as you can see here. So the flow is coming from the right side of your screen, and the particles goes, so they make this, make plus here and here. Okay, so this is very interesting, and because, well, also during many years we were wondering how this animal could feed and the feeding habit of this animal. So, well, we have some clue about how was possible to do this. So, okay. With regard to the leaf coefficient, the simulation shows a very low values of the leaf, and actually the leaf coefficient is oscillating around 0, and this is super cool and very interesting. So if we look at the superficial pressures, they are very negative values in the dorsal side, and in the ventral side, and only slightly positive values in the snout here. Here, and this is quite interesting and supports what we have seen in the velocity fields diagram. So microparia was very stable when the trilobite was in this swimming. With regard to, well, this is the rear flow, and with regard to the animal, when the animal was enrolled, this is also very interesting and still very stable, but we have other things that worth to mention. And it's very stable, and again, fluctuation around 0, but very, very, very stable fluctuation. This is the red line, okay? And well, we have this positive pressure here in this position, which is pushing the trilobite up. It's the same pressure that we have seen in the velocity lines, but this balance with this blue section here and this dark blue. So the trilobite really was very stable. So the results are suggesting that probably the trilobite was able to be in the water column without any movement independently of the velocity, or at least in the range of the velocity that we have analyzed. So we have something like this. So when the trilobite is enrolled, so we were wondering the meaning of enrollment in a water column, and well, the trilobite was like this, so probably some kind of balance. So we are wondering if this is a protection against a predator or storms, or just is a cheap and very easy way to travel, or resting in the water column, and they could be in the water column. And of course, any other idea is welcome. So, well, just to conclude some discussion and conclusions, the available data suggests that the nekton revolution was gradual throughout the lower Paleozoic and first steps during the Cambrian and new important wave during the Ordovician, that I think we have to explore new ways to assess the transition between benthic and eunectonic animals to understand the mechanisms of biodiversity during the lower Paleozoic. So this is something that we have to face in the following years. Ordovician encyclopedias have already the adaptation, so in the way they are already adapted, so how they arrive, how they change, how forms change in the upper Ordovician, lower Ordovician, and it will be really nice to analyze other forms. We will do it in a future, and it's very nice to see that the aplesiomorphic behavior like enrollment was a very nice adaptation for a new habitat in trilobites. So we know very well that trilobites enroll from the lower Cambrian, and when they suddenly arrive to the water column, they are still using, we don't really know for what, but they still using and seems very, at least very effective in the water column. Well, this is my talk. Thank you very much for your attention, and questions and suggestions are welcome. Thank you.
Great, thank you so much, Jorge. That's a great talk. I pretty much enjoy the animations and models. That's going to be exciting for my intro classes.
Well, I have to say that I didn't really enjoy this talk. This is, guys, I really missed you. I would like to stay in a real meeting next year. Yeah. Cross fingers. This is very weird to talk to the screen.
Yep. Welcome to 2020.
Welcome. Yes. Where is my I don't know if you can see.
Few questions we have in the chat. So Javier, you have a question. Do you want to ask that, or I guess I could ask it for you? He asks, he would imagine that the trilobites would need to keep moving their limbs to maintain buoyancy within the water column. Wouldn't they sink right away once they enrolled?
This is Javier question. Yeah. Yeah, good question. Well, of course, for this analysis, we remove all the legs. So probably, we have two different things. On one side, the trilobite was just not swimming, just was against the current or in the same way of the current. And on the other side, probably they move some metacaudal wave to swim with the legs. We have many animals today, many arthropods that they have multi legs, and they swim and they use it for swimming. And the other question, sorry, what happened when it's enrolled?
Yeah, wouldn't it sink right away if it couldn't maintain the buoyancy?
I don't know.
Can I actually answer directly?
Sure.
So, hey Jorge, great to see you.
Good to see you too.
I love the animation. That was fantastic. So I mean, what I was thinking is like, the question is like, I would imagine they would have to keep beating.
Sorry, Javier, I cannot hear you very well.
Okay, got it. Is that better now? Is that better?
Slightly better.
Okay.
Slightly better.
I would imagine they would have to keep beating their legs to keep moving. So
yeah.
If they were to enroll in the water column, I mean, maybe that was some sort of escape behavior, because they would sink, I would imagine, quite rapidly. So they could get away.
But I didn't explain very well. So the point is that this trilobite, when it's enrolled, doesn't sink. So that's the interesting thing. So it's like a drop in the middle of the water.
Mm-hmm.
So when it's enrolled, it's just floating and nothing else. So it's not going to sink. You know what I mean?
Yeah.
Because the hydrodynamics prevent sinking. So we have a pressure up here and another here, but it's imbalance. That's what they want to show in that reconstruction, that the trilobite is enrolled, and when it's enrolled, it's balanced.
Mm-hmm.
And so some kind of protection.
But then, I mean, that would also depend on the speed of the current around it, right?
Yeah,
I mean, if it's in a strong current, it would stay afloat or buoyant. But then if it's in a very still water, I mean, it would have to be very, very tiny to just stay with the viscosity of water, I guess.
Yeah, but if I don't know if you realize, so we analyze from very, very low velocities up to 2.5 meters per second, which is really high velocity actually. So it's not an hurricane or a typhoon or something like that, but it's high enough. And still the stability is unbelievable. It's always around zero.
Okay.
We can talk later.
Of course, yeah. Well, awesome. Thank you so much.
All right. I think, Christian, I don't know how quickly you want to jump into the coffee break, but I guess I'll just end with Sophia's quick question. she may have missed a part, but wondering if you suggest that Placoparia is nectobenthic.
Sorry?
Sophia wanted to know if Placoparia is nectobenthic. Was that one of the suggestions?
No, for me, you know, Placoparia, so the hydrodynamics is suggesting that the Placoparia could be, have some kind of hopping locomotion. But nothing else. No, because the drag coefficient is very high. So the key question here is the drag coefficient, and the drag coefficient is too high to be nectobenthic. It's almost impossible.
Escape me. I'm sorry, I just changed the language. Hi, Jorge. And because I thought I was reading that suggesting that it was mesopelagic or something. Because it's blind. It makes no sense. Okay.
Microparia. Oh, sorry. Microparia. I misunderstood. Microparia is a mesopelagic trilobite. It's very clear. But Placoparia is a benthic.
Okay. Yeah. Okay. I just missed that part, and it was looking.
Okay.
It
yeah.
seems.
Sorry, I misunderstood.
Okay. Thank you.
No worries.
Okay, great. Well, thank you, Jorge, for another.
Thank you for your invitation.
Yeah, and I want to thank everybody else for all of our speakers and guests. We've had some great talks for the early session. So thank you to everybody. And then, yeah, I guess, Christian, I guess you can step up to the plate here and tell us how you want to handle the transition here.
I can do that. I think we're just going to create the breakout rooms once again, and then reconvene in, 15 minutes or so that it's 1/2 past something, wherever you are.